Hi guys, if you're joining me. Welcome, welcome, welcome. If you have any questions, please feel free to ask. How are we doing? Welcome. If you have questions, please do ask away. Hi, how are you doing? Nice to see you too. I'm just waiting on my group tuition video downloading, so I thought I would jump on here whilst I wait. And I would, yeah. Hi. Answer any questions that you may have. Hi, Ari. Thank you for the likes. Do you know you're one of my, what was it say, 10%, top 10% of your top 10% viewer? Habibi, you are up there. So thank you very much. I appreciate you joining the live and liking it as well. It does help. So what is it that you would like to cover today? Specifically anything? You deserve better, not gonna lie, you're literally... Oh, thank you. I have a question about single circulation. Go for it, Ari. What's your question? Thank you. It's really lovely to be appreciated. It really is. But what's your question about single circulatory system? So you're talking about the fish, I'm guessing. Do we have to know how it works within a certain organism? So, Ari, which exam board are you doing? Are you OCR or are you AQA? Because with OCR, you should know it for OCR. So you should know it for the fish specifically. However, they have thrown in a question where they gave a fish, an amphibian, and a mammal, and they wanted you to compare the similarities and the differences between those different organisms based on what you could see in the insert that was in the exam. So you should know about the fish and how they have two sets of capillaries, and you should know about they have one ventricle and one atria rather than the two things like that. Could you teach Educus Biology A-level? Yes, I can teach. I can teach all the exam boards basically as long as I have access to the specification and I have access to the past paper questions. I can teach them all. I wouldn't say that I know it off the top of my head. Like if somebody asks me about OCR A, then I can tell you which part of the specification, what it is specifically, because I'm an expert in that. And I've built my channel around OCR A because there wasn't anything for my students. But with the other exam boards, then yes, I can teach it. I would need to do more prep for teaching it. So I tutor one-to-ones with different students who do sit different exam boards, but it's about what they require and it's about me doing the work. Now, I wouldn't be saying that I can just tell you everything right now off the top of my head, so I would go and look it up. How would you recommend revising biology A-level if I find it so hard and feel like the learning of it? Is this UK? Yes, it is UK, and doing questions is so different. So, okay, when it comes to A-level biology, it is a tough subject. So a lot of students pick it because they think it's going to be the easy of the sciences, and it means they can then go on and do a STEM subject later on. But it's actually really tough because there is so much content to learn. Not only is there a lot of content for you to learn and understand, it's the level of detail you need to go into. So what would be my advice is start with the basics, start with the foundation skills. And what I mean by that is understand the key components. So I've just been doing tonight osmoregulation with my students. And to understand osmoregulation, you have to understand the membrane structure and the fact that different cells in different areas within the kidney nephron have got different proteins inside them, and that therefore is going to allow different molecules to move in different directions depending on concentration gradients. So biological membranes, you need to really, really get your head around that one and know it. Biological molecules is always one that you need to know because it's so important through all the different parts. However, a lot of students get hung up on that. They get hung up on the fact that they need to learn the biological molecules and that's all they revise, and they think it's just going to be about that, but it's not. So I would say nucleic acid and nucleotides, get that under your belt, understand it, not just read it and think you know it. And that's the thing, when you say the questions are different and they're difficult, that's because they're assessing whether you really know the content. Every time I do my sessions with my students, so I have a student who I had today, earlier today before my group sessions, and when I met with that student and we had said, right, we've covered that content, so we were doing cellular, what were we doing? We were doing manipulating genomes. So we just finished all the manipulating genomes. And I said, okay, do you want to do something else, or do you want to do some questions towards it? And we did start with some questions towards it. And what was really interesting is when we started doing the questions, it was a way in which we were identifying the missing areas within their knowledge. And that's basically what I do. In my sessions, I can just keep talking and tell you all the content. That's what my videos are for on my channel. Basically me going blah, 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 and telling you everything. What I do in my sessions with my students, and what I would advise you to do yourself, is I ask lots of questions and I try to figure out what you don't know, and I ask questions that are not just about what's in front of you in the exam question. So if I've got an exam question, I'll start asking, taking us off in a little tangent, because I'm trying to figure out what you do and you don't know. Because if you can't answer a question that I've asked you, so we were talking about stem cells and them differentiating, and what the student was not quite getting, or no, that's not correct. What they weren't remembering was the fact that the stem cells need to go through mitotic division before they can then differentiate. Well, what needs to happen for mitotic division is we need to take a step back and remind ourselves about interphase. And so therefore we need to remind ourselves about G1S, DNA replication. So if we were looking at, which we were, gene therapy, and we've inserted a gene because we've done somatic cell gene therapy, if we've inserted a gene, that cell's not going to go through mitotic division because it is a cell which has differentiated, and therefore it's in G0. So because of that, if there's any damage, the cells that are going to replace it are going to be those with a faulty gene. And so because of that, we were identifying these little bits of information that they just forgotten because they haven't done it for such a long time. And so that's what you need to kind of work through. So the those mark schemes, the SNAB ones, I actually, do you know what, it's a funny one. I tutored that a number of years ago, and I would say that they are one of the really specific mark schemes when it comes to looking at different exam boards. But the content you have to learn isn't as much, but it does have to be very specific, the answers that you're giving to the question. Is this OCR A? Yes, it is. So if you have questions, basically I answer any questions that people come on and ask when I'm on here. I have basically been I'm waiting on a video downloading. So I am jumping on here whilst I'm free to do that. And I'm just checking if I have missed any questions. And if I have, I'm so sorry. You're in year 13. Would you recommend me do lots and lots of... Yeah, absolutely, 100%. you need to be doing as many past papers as possible. And the reason being, the more past papers you have exposure to, the more likely you are to see patterns in the mark schemes. And when I say that, I don't mean memorize those mark schemes. See the patterns in the mark schemes. And also using, for example, if you're doing the multiple choice sections of paper 1, paper 2, what you can do is when it's got a question where the multiple choice, you've got to get the right answer. If you can't eliminate the wrong answers, you don't know how to eliminate the wrong answers, you don't know your content. And that's one of the key things I would say. Using multiple choice questions is really good for revision, to help you figure out why you have gaps or what gaps you have, because you don't know how to eliminate those wrong answers. So yeah, absolutely, 100%. As many questions as you can is going to make a big difference. And also, when you do the past paper questions, when you're practicing these questions, what's important is, a lot of you, again, what you're going to do is you read the question, you answer the question, you look at the mark scheme, you tick or you cross it, and then you look at what it says. You either rewrite that word for word, or you just think, I'll memorize it and move on. That is not a good use of time, and that's not going to help you. It's important what you're actually doing is that you're reading that mark scheme, you're figuring out what bits you don't understand of the mark scheme. If you read that mark scheme and you can't understand why that's the answer, you don't get what they've written, you're not going to be able to apply that knowledge to another question. So it is important that you are not just reading mark schemes, that you're not just writing the answers, checking and marking and be like, oh yeah, I got 5 out of 105. Well, it's not obviously 105, but you get what I mean. It's important that you are actually analysing what you've done. So every test paper you've ever had back, what you should be doing is looking on there to see how many marks you have lost, where you lost the marks, what caused you to lose the marks. Was it, for example, the detail? So many students, and this is so frustrating and also upsetting for students, in the fact that a lot of students still, to this day, even though I keep saying still only give GCSE answers, the detail you need to go into for A level with your answers to get the high grades is keywords. If you don't know your keywords for the topic area, you're not going to get the high marks. And that doesn't matter whether you're AQA, whether you're OCR, whether you're EdXLB, whether you are the IB. It doesn't matter which exam board you're sitting. If you don't know your keywords, if you don't know your detail, you're not going to be able to answer the questions to the level that needs to get the high grades. So students are shocked when they end up getting a grade which is a C, for example, when they came in with 9s at GCSE. They're like, but I'm a 9 candidate. I got 9s in my GCSEs, straight 9s. And it's like, yeah, but that was GCSEs. GCSE and A level are different. Just like A level and first year university is very different. Each time you're adding a layer to your knowledge and understanding so that you can then become an expert in it. And that's tough. And it's meant to be tough. It's meant to be challenging. It's a level 3 qualification. It's not a level 2, which is your GCSEs. It's a level 3 qualification. So you have to get that knowledge. You have to be able to show that. And that's the bit that's missing. And it's frustrating. And that was the one thing I would say, that a lot of students still only give GCSE level answers because they think that's enough. Like photosynthesis and respiration, you've learned about it at GCSE, so you've learned more in A level. Can you give more? Inheritance, you learn about inheritance at GCSE, you learn about dominant, recessive, codominant, but can you then go into the detail that's required about what does it mean for that expression? Can you talk about transcription, translation? Can you talk about the codon, the anticodon? Can you talk about the hydrolysis of a molecule? Can you talk about the condensation reaction that builds a molecule? Do you have those keywords to help you get the higher grades? I went on a massive rant there. I don't even know where that came from. But yeah, that would be my bit of advice if I am giving you any advice tonight. It's one of those things, it's hard. It's very hard because you want to be giving the right answers. You've done the revision, but you've also got the other subjects to remember as well. And it's maintaining it within your brain and then knowing how to retrieve it later on that's going to make the difference. Retrieval is the hardest things. We did gene expression prokaryotes. Oh, nice. So you were doing the lac-operon, I'm assuming. So I'm assuming you've been looking at your LAC-I gene and your repressor proteins and your operator and your you're welcome. I'm glad that you like the advice. It's one of those things that everything I do on this channel, everything I do is just basically me giving you my experience. Lactose, yeah, lac-operon, exactly. Can you go over gas exchange in an insect? Yeah, sure. So interestingly, Ari, you talked about the single circulatory system earlier, but now you're asking about the gas exchange. Now, the insects have an open circulatory system, and because they have an open circulatory system, they have to have a completely different gaseous system because they deliver their gas directly to the cells rather than the gas being taken in the bloodstream and circulating around. So because they have a different system, their system has got these openings that run all the way along the side of the body, which are your spiracles. And your spiracles then lead into your tracheae. And that's an interesting one because if we're talking about the trachea here, it's spelt slightly different when you talk about it in the insects. It's got an E at the end. So we have these spiracles, which are these openings, and these openings actually have sphincters, and these sphincters can close and can open. So that's also important. But they have these openings all the way along the side of the body, and then that leads into the tracheae, and then that leads into the tracheoles. So basically what we're doing is we're taking a really long tube and we're branching down into smaller tubes and smaller tubes and smaller tubes. So the tracheoles are these smaller branching tubes which lead in amongst every cell. So if I'm trying to think of how to do this. So if this hand's my cells, then the tracheoles kind of lead in all the way through all the cells. So what you're going to get is the diffusion of the gases going directly to the cells rather than going in the bloodstream. So the key thing when it comes to the insects is they have their exoskeleton on the outside, which is going to prevent gases from getting straight into every single cells. So they had to evolve an exchange system, which is the tracheal system. And so therefore you've got your spiracles, openings all along the body, the tracheae, which are these tube-like openings which are maintained open. So our trachea is open with our cartilage rings. The tracheae are opened with the chitinous rings. So it's a different polysaccharide material which is keeping it open because we've got a different organism, different molecule. And then that leads into the tracheoles, and we require the diffusion, the gases to diffuse. So they need a way of ventilating it. So they need a way of, for example, if they're an insect that does a lot of flying and they need a high supply of oxygen because to meet their metabolic demand, they need a way of ensuring that movement occurs. So they can do a process of ventilation like we can. We ventilate, we open and close our rib cage. Open and close, that's not even the right word. We lift our rib cage up and out, and we bring our rib cage down and in, and by doing that we change the volume and the pressure within our thoracic cavity. So therefore that's going to push or allow the air to flood in. So with them, if they are a insect that has a really high metabolic demand, they're therefore potentially going to do a thing called muscular pumping. So you'll see this with like locusts or grasshoppers, where they will do a muscular pumping, which is going to ventilate and make sure that there's more air being supplied. They can have a collapsible sac, some of them. So sometimes they do show in a picture that they'll have like a sac that they can fill up for a reserve of gases for that when it's needed, that they can collapse down. So it really just depends where the question's going to go, but I hope that helps to answer that one. You learn the lactose specifically is the lac operon, because that's the one you need to know. I just saw something. I understand that part. I don't get the what. Right, okay. So the water filled, the tracheal fluid, and I saw someone else say, what's the purpose of the tracheal fluid? So as I'm talking right now, I'm talking away to you, and what I can feel happening is the more I talk, the drier my mouth becomes. We lose water vapor through openings. So because we lose water vapor through openings, we need to ensure that there is fluid that stops our cells from drying out. So the tracheal fluid is there as a way of preventing drying out of cells. So making sure that the cells don't dry out, making sure that there's a fluid there. However, that fluid actually acts a bit like a barrier. And so what you can get is, if there's too much fluid in the area, it's harder for the oxygen to dissolve into, and that becomes more saturated. So therefore the whole lactic acid, what that does is that actually results in there being less of the tracheal fluid, which therefore results in there being more exposure of the surface of the tracheals for the exchange to happen. So it's kind of it's there as a necessity, but it can also be a hindrance. So it's about the way in which it's used together. Paul, how are you? Very nice to see you on here. What was cyclic AMP again? So cyclic AMP is a secondary messenger inside a cell. So if you have a hormone which is a non-steroid hormone, so if you've got a hormone that cannot get across the phospholipid bilayer, I was actually talking about that in my group tuition session tonight, because we were talking about ADH. So if I use ADH as an example, antidiuretic hormone, ADH, is a hormone which is a non-steroid hormone, which means it cannot cross the phospholipid bilayer. So instead it needs to bind to a receptor which is on the surface of the membrane. So if it's binding to a receptor on the surface of the membrane, we class that as a first messenger. So that hormone has a message. That message is going to signal to the cell that something needs to happen within the cell. Now, it can't cross the barrier, so what it does is the ADH binds to the receptor, and that causes a cascade of events to occur. And that cascade of events results in a secondary messenger. That secondary messenger is cyclic AMP. So when you have your adenylate cyclase, which is an enzyme, this enzyme becomes activated, and with that activation, ATP gets hydrolyzed down into cyclic AMP, adenosine monophosphate. Now that adenosine monophosphate, that cyclic adenosine monophosphate, that will then go and phosphorylate a protein kinase. And a protein kinase is a type of enzyme inside the cell which will then activate something else. So if we were talking about adrenaline, adrenaline is going to, adrenaline is going to bind to the adrenal receptors. Antidiuretic hormones is going to bind to the ADH receptors. Whatever we're talking about here, we're talking about a hormone binding to a receptor, first messenger, that's then going to, activate the adenylate cyclase. The adenylate cyclase, that secondary messenger, then activates protein kinase. So as I said, with the ADH, that protein kinase results in some vesicles containing aquaporins fusing with the membrane. If we switch it over, we talk back at adrenaline, with adrenaline, thank you so much, by the way, for all the gifts, that's really lovely. With the adrenaline, what we're going to activate, for example, is glycogenolysis. So the protein kinase, everything is still the same. Adrenaline this time binds the receptor, that then activates the adenylate cyclase. The adenylate cyclase converts the ATP to cyclic AMP. The cyclic AMP is then going to activate your protein kinases, but this time the protein kinases are to do with activating enzymes that are needed to break the glycogen down into glucose, and so hydrolyzing the glycosidic bonds. So cyclic AMP is this secondary messenger which then activates others. Now, somebody was on here earlier saying that they've just done the lacoperon, which means they're doing cellular control, and that therefore means they're going to be doing the regulation of genes. So if we use the cyclic AMP as an example, it's a really good example for post-translational regulation. So an enzyme has been translated. So we have the transcription and translation producing the enzyme. So because we've got this production of the enzyme, it has its primary structure, the sequence of amino acids. It has its secondary structure. Is it the alpha helix or is it the beta plated sheets? And then you have the tertiary structure, which is determined by the primary structure. Now, if it's an inactive protein, that inactive protein is regulated by the presence of the protein kinases or the cyclic AMP. So with protein kinase is activated when the cyclic AMP binds to it, changes its shape, allows it to do the job that it needs to do. And so therefore that would be a protein has been translated. We're going to regulate it post-translational. It only becomes the right shape and complementary to the substrate it's going to work on when there has been a signal for it to occur. We're regulating it at the post-translational level. So we don't need to translate the protein and then have it do the cascade of events, because that would be a transcriptional level one. This is that the protein's sitting ready waiting, but it's not activated until there is a signal. In this case, we have the ADH binding, then activating for the secondary messenger, cyclic AMP, and it goes from there. I hope that made sense. That was a lot of rambling from me. Speeds up the transcription, and that oxygen reduces the products of cyclic AMP. It's a really interesting one. Oh, for when the blood. Oh, did I miss something? Hold on. Oh, thank you. I do appreciate. Lots of different things you sent as well. That's crazy. Is it the animal response or hormones topic? So interestingly, it's all different ones. So what I've just done there is adrenaline is animal response. Cyclic AMP would be the adrenaline fight or flight response, that side of things. The antidiuretic hormone is excretion. The steroid or non-steroid hormone is the hormonal system. So it basically depends where they want to go on the question as to what it is that they're looking for. Is it really hard to get an A star? It is tough to get an A star. It is possible. However, it is that you have to work at it and you have to keep going as you go. Year 12 is really important at getting that foundational skills down. Sorry, I missed a whole load of questions. I'm just trying to work my way back. So, is glycogen made from monosaccharides, glucose, fructose, and galactose? No, glycogen is made from lots of alpha glucose molecules with 1 to 4 and 1 to 6 glycosidic bonds. Well done, Ari. It is. Yep, you're absolutely right. Alpha glucose, which is branched. Yeah, and it's really important that you say the 1 to 4 and 1 to 6. So when you say it's branched, that makes it sound like it's just the 1 to 6, and you want to say 1 to 4 and 1 to 6, which gives it its branched appearance. Sorry if I've missed your questions. I am trying. Chinema, how are you? I want to send a mixture. I love it. Thank you very much. Of my 21 pence that I've earned on my TikTok account, you're probably the person who's paid 20 pence of it. So thank you. I appreciate that. Oh, mocks are over. Woohoo! Oh, well done. You got a B plus. A B plus is positive. Oh, I'm sorry, Chidima. I'm sorry about that one. Your grades fluctuate. So the key question I would say for you is grades will always fluctuate, so I'm not worried about that. That I would expect. There'll be topics that you find a lot easier than other topic areas, and also it depends how they've assessed you and what they've used. What I would say is, do you know what it is you're doing that's holding you back? So have you asked your teacher how you can improve? What have they actually given as your specific example for what you need to do? Not a generic answer, which is quite an easy one, but to give as teachers. What I would say is, if a student came to me and said, do you know what, I really need some help, what is it that I specifically need to do? Now, this isn't, I'm not saying in the class, I'm not saying disrupt the lesson. What I'm saying is go and find them at another point and say, look, I really want to improve. Can you give me any guidance? What is it that I need to do? So that's the first thing, is identifying where you're losing the marking points. What is it that's holding you back from getting those A to A star grades? Now, for me, I know that my math skills are ones that I have to work on, and I know that I have to take the time to really, it takes me such a long time to do maths. It's crazy. It should not take this long, but it does. And I have to double check, and sometimes I'll put all the numbers correct in the calculator, and then I'll write the complete wrong answer. I know that I do that. So because I know that I do that, I then go back and I double check my answers quite often. So it's about figuring out what it is you're doing wrong. Which topic areas are you struggling with, and what, with the time you've got left, what is it you need to focus on? So for example, in all the weeks that we've got leading up to the exam, I know with my group tuition sessions that I don't have a lot of time. I have 20 weeks, not even 20 weeks left. We're down to what, about 18 weeks now, up to the May half term. So what that tells me is I need to be selective on what I do with the students in the session. And sometimes I want to throw so much into them, and I feel bad. Like tonight, the students were a bit overwhelmed because I was trying to get so much crammed in with still giving time for questions. And this is what you have to do. You're never going to be able to do all of it. As much as you think you can study all of it, you're never going to be able to study all of it from now until the exam to the depth that you would want to. And you don't need to. What you need to do is you need to figure out, what don't you know? What topics do you hate? What do you pray never comes up? And work on those ones. As much as it pains you, as boring as you find it, as hard as it is, do that content again and again and again until it sticks. Because if you go and spend the time doing the stuff you feel really good about and the questions that you really like because you know it, all you're doing is you're wasting that time. And that's hard. That's so hard. Don't worry about, you're not spamming, you're asking questions, which is basically why I do these sessions. So do not apologize. If you didn't ask the questions, I wouldn't have anything to talk about. It's not like my sessions where I plan to, like, every minute of every, I'm like watching it going, oh, I don't have enough time to finish this. It's not like that. This is just, I pop on here and I help any questions that students have. So please don't ever apologize for asking questions, because that's what it's about. I'm working on flashcards that have helped with my memory so far. Yeah, so a lot of active recall is really positive. But one thing I would say, like, doing things like this, I have to say, recording videos of yourself answering questions or having a video chat with a friend who's like a study buddy. You don't have to be in the same room as each other, but you could be doing right, we're going to do some questions and I'm going to ask you, you're going to ask me, and then listening to the answers back, listening to what you say. I mean, it's so cringeworthy. Like, social media is one of those things that I'm like, oh, how is it I am doing this? Because I'm so cringy with it. Is it possible to go from a D to an A in 5 months? Absolutely, it is possible. However, the amount of work you would have to put in to get that increase, it's absolutely possible, but it's about the effort of what you would need to do. So your key thing is identifying what you are losing the marks for? What's holding you back? What is it you're going to do you have the time to dedicate, the motivation? Nobody can stop you. Absolutely nobody can stop you from achieving where you want to get to if you have the time, the motivation, and the effort to get to where it may be a slog. And I'm not going to lie to you, it's not going to be easy. And there will be people around you who just get it. And you're like, I must be so stupid because they get it and I don't. That's not true. Their brain just works differently from you. You need to figure out what you have to do to get it to stick, to understand the bits that you didn't know, and then learn how to answer the questions. And it's the answering the questions. And so therefore, I mean, I don't know if you have the resources available, but if you are wanting to work from a D to an A, I would suggest, if you can, getting and sourcing a tutor to guide you specifically on what you need, because you've got such a big jump to do. What is it that's holding you back? So I would say, if you have the resources, then tutoring would be something that I would suggest. I would say potentially one-to-one to gain that knowledge of where you're losing the gaps to begin with, and then moving potentially to groups. But it is possible, but it's about whether or not you have the time, have the resources, and can work for it. But it's absolutely 100% within your grasp. I would never have said that when I was your age. I would have been like, no, it's not possible, because I didn't believe it. I didn't believe that I was able to do the things that I can do now. And I think that held me back for so many years. Because for those of you who've only just come across me and don't know my story and my background, I got a B grade in year 12 equivalent. So obviously I'm from Scotland. And so I got a B grade, and I only did year 12. I didn't do year 13. So I left school. It wasn't for me. I didn't like learning. I didn't like education. I was really stupid. And I got a B and 3 C's. So I got English, maths, biology, and PE, so physical education. And good luck for the Oxford offer day. So I did those 4 for year 12, and then I left school and I didn't stay on. And then I took a year out, worked for a year, went to college for a year, got a higher national certificate, got bored of that, didn't stay on, dropped out again. And I didn't go to uni until I was classed as a mature student when I was 20. And because I had more life experience, I was different, I was ready to get into learning, I went to university. And when I went to university, I did biology as my degree, and I had to relearn a lot of the stuff that I hadn't remembered or maybe didn't learn. And I spent 4 years of university to get my 2-1, and then I came to Cambridge. Now, if you'd have said to me when I was your age and I was sitting there that I'd be someone who's got not only a postgrad in science teaching, a postgrad in restorative approaches, and also a master's of education from Cambridge University, I would never have believed you. I did not think I was smart enough. I did not think I was able to get the grades necessary to be successful. But what was holding me back was my belief in myself. I didn't understand how my brain worked. I didn't understand how I needed to push past the struggles. What I did know is other people around me just got things really easily and I didn't. So I must be really stupid. So what I'm saying to you is, it depends on whether you have the resources, the support, the motivation, and the effort. But the only person who will hold you back is you, if you really, really want to get whatever it is you want to get. That's my little motivational speech for you. But that is honestly, I mean, when I wrote my thesis, I started my thesis off on the fact of your self-concept, your belief in yourself, and how that can hold you back. I have parents who left school at the age of 16 who both went off to the armed forces, and that was how they had their careers. And so I am the only one of my family. I've got a brother and a sister, and I'm the only one who's actually been to university in my family. And I was the one who dropped out of school and didn't continue to year 13. So it's a funny thing in here that when someone says to you, for example, as a parent, I say, I'm not very good at maths, And I can also say to my children, well, you're not very good at maths because I'm not very good at maths. That could actually instill in my child that they're not good at maths, and actually it's not. I just know that I've got struggles. I know I have to work harder. My brain just doesn't see the numbers properly. And so therefore I have to work at that, and therefore I need to guide them on how to do it. It's an interesting one because I completely agree with you in the fact that some people can help you and motivate you even though you might feel secure around them, but it's an interesting one of, it's a lot of the time down to confidence and down to people can fake it. I said to my daughter on the way to school today about faking it, and she was like, I can't fake it, mummy. And I was like, well, you can. It's just hard to fake it, but you can fake it and practice until you get good at something. And it's an interesting one. Some people are just really confident in the way that they give an answer or the way that they display themselves. And actually, what they're saying is a load of rubbish. Or they don't actually know it. Now, don't get me wrong, there are people out there that do know it and are very confident as well. So they're on a different level. But sometimes it's hard, but sometimes actually you have to question what other people are saying because they sound like they know it, but actually they don't, or they only surface level know it, and they can't delve into the nitty-gritty parts of it because they don't fully understand it. And actually there'll be bits that you get that they maybe don't, or they're not as strong at. And so it's about finding where your strengths are and your passions are. I'm sorry guys if I've missed a whole load here. I do apologize. Oh, thank you. I'm glad that you find it an inspiration. It's a funny one. You're saying it's an inspirational story, but that is just my story. That is what I did. That is how my life changed. Like, I was basically a school refuser. Like, it's funny, when I was in primary 4, so that would have been year 4. It's a funny story we say now in my family, but would have been year 4. So what age is that? Year 4 would have been about 8, 9. I bunked off school. I literally left school. It was very different in those days, but I told them that I wasn't very sick and that my mum was at home. And they let me leave, and they let me go home, and I went home and I let myself in the house, and so I walked all the way from school home on my own. I got my 2 dogs out and I took them for a walk because I didn't want to go to school. I didn't enjoy school. And so at the age of 8, I was walking around the streets with my dogs, and my gran and grandpa drove by me, and I had to jump into some bushes to hide away from the fact that they were going to see me with the dogs. Now, they didn't see me. It's a story that my parents are aware of. We've talked about it. It's like a funny story now because I'm 43. But at the time, like, I was a school refuser because I really didn't believe I could learn. And so it's so amazing how your life could change based on, one, who you surround yourself around. Like, I used to have people who were much smarter. Even my brother like just gets things really easy, or he looks like he gets it really easy. And because he looks like he gets things really easily, in comparison to him, I struggle. So I'm not very smart compared to him. Now, he's got different strengths and he's often running his own business and he's very good at what he's doing. But he got straight A's, he went to university and then dropped out. That wasn't for him. That wasn't the right path. And this is the difficult thing about one school, about the path that you take, that sometimes you feel like your path is from A to B, but actually you might need to go from A to D to F to G to C to E to, and then on to get to B. And so taking all these different routes could still lead you to that end place. It just might not be the same path as someone who went from A to B. And that doesn't make it any better or any less than you. It's just different. And that's hard. It's really hard knowing that there is so many different ways and different things that you can do in life, and so much pressure. Never give up. That's amazing. And that, do you know what? That's really positive. So you had to retake your English GCSE 4 times to finally pass. And that's the thing, because you persevere, because you didn't give up, it makes it so different. And that's the thing, if you're doing something that you enjoy, like if you're a footballer, I'm just going to go down this route. If you're a footballer or a dancer, or you are a dress designer, it doesn't matter what it is your passion. When you're passionate about something, when you like something, when you want to do it, you're willing to spend the time, and hours can go by and you've been spending so much time doing something that you really love and you're passionate about. And so therefore you get better at it the more you do it. So I've been practicing learning how to draw, and I've been doing little bits, and I had so much fun over the Christmas period actually learning to draw. And these little skills that I've been learning, now I've tried them and I've been doing them, and it was amazing. And the more, when I look back from a year ago when I first started drawing to what I've done this Christmas, I'm like, what a massive difference. But you don't see that when you are doing something on a regular basis that you love. You don't see the effort you're putting in, because you're like, it's just what I do. This is it. This is my, I'm doing what I love. It doesn't feel like hard work because I'm interested. Now, if you saw me yesterday on my live when I was trying to do the dishes, I absolutely hate that. I hate doing the dishes. And so because I hate doing the dishes and I hate cleaning and tidying, it feels like I'm being tortured every single time I do it. I don't want to do it. And so it makes it so much harder. So what I would say to you is, everything you are doing is about taking those steps forward in the right direction to make progress. And the one thing, Callum was on my live last night. I can see you there, Callum, because we were chatting about your thesis, which I'm excited to hear more about when you do it. But the thing is, when you do something you love, like I could spend hours building the resources that I spent. Actually, I did spend hours. I started at 9:30 this morning, and I had a client at 2, and I spent until 1:30. So 9:30, 10, 11, 12. Yeah, so 4 hours planning for my 1 hour group tuition session tonight. So I spent 4 hours. Now, I didn't feel like I was spending 4 hours. I was doing things that I loved. I was doing things that I really enjoy. I was tweaking. I was making it better. I was improving things. And so that's what I love. And so that felt like I spent 10 minutes on that, and I didn't spend 4 hours. Now, I had to do the dishes yesterday, and for those of you who were with me, was maybe 15 minutes. And that was torture. If you guys hadn't joined me, I would have struggled and just been like, oh, I don't want to do it. I just hate doing the things that I really don't want to do. And so I guess what my rant is all about is that appreciate when you make the time and you put the effort in and you're actually working towards something, that it will be moving you in the right directions, even if you feel like you're taking 10 steps backwards. Every bit of attempt that you do will take you a step closer to where you want to be. And I mean that when I say as well, trying to answer questions, it really frustrates me. It's one of the biggest things that I hate, when students don't attempt an answer because they worry that it will make them seem stupid, or that I will judge them because they haven't given an answer to a question that I've asked. And that's not what frustrates me. What frustrates me is the fact that because you haven't put anything down, I can't help you. And so your teachers want you to attempt. And the more you attempt, the more you finesse, the more you improve, the more progress you will make. The students who don't make the progress, the students who struggle the most, are those who are scared to put things down. Things you were praying for years ago. I didn't think. And this is all about the amount of work and effort you're going to put into to get you to where you want to be. But it might not be first time. It took me 3 attempts to pass my genetics exam in my 3rd year of uni. If I hadn't of completed it the final time, I would have been kicked out in my 3rd year of uni. But luckily I passed. 41%. I only needed 40% to pass. I got 41%. I passed. I was able to move on, and now look at me. Things change, paths change. I mean, I went from being a teacher to a head of biology to a deputy head of lower skill to a head of house to a director of student progress. I then had my girls, and things changed. My life changed, and now I am here doing what I do now because of how my life has changed, and that's for the better. I had it in my head that there was one path I was going to take, and then life happened, and I have gone in lots of different ways. I'm now learning new skills. I make all my diagrams in all my videos, all my diagrams in my study guides are ones that I've learned how to do on my own through using Adobe Illustrator. My YouTube channel, I mean, I'm still not utilizing it enough, but I've done all this all on my own, all skills that I've learned from just watching other YouTube channels. And so I hope that the stuff that I put out there is helping you guys as much as I want it to, because I've got 20 years experience of teaching. I love teaching. I love helping students. And so I want to ensure that you guys are getting the knowledge and the experience that you deserve and access to it. Just because I'm not in it, and I'm not trying to big myself up here, but just because I'm not in the classroom anymore doesn't mean I shouldn't be able to help lots of students. And so that's why I do what I do. So I really appreciate all of you who come on here, who ask me questions, who watch my videos, who like and share my videos. I really appreciate that, by the way. And I will be back on here because I've just noticed it's 10 o'clock. I will be back on here on Wednesday. I'll actually do some actual teaching. I've had my slide open and I haven't written anything at all on my PowerPoint, but thank you. There you go. There's my little thank you to everybody who's been with me tonight. Guys, have a good evening. Good luck for those of you who have got the Oxford Offer Day. I wish you all the best, but I will be back on Wednesday at 9 o'clock after I've done all my sessions there. Thank you. Thank you, guys.